Thank you.
Okay, thank you. Thank you everyone for having me here. It's a nice opportunity to share some background about my research. Today, I'm going to talk more about the research I have done during my PhD back in Brazil. So this topic I want to talk to you today is how we can use Enviromics to increase the accuracy of genomic prediction for multiple environments. And for doing this, I'm going to show you some results in tropical maize back in Brazil and some tools I have developed. About my background, I'm from Brazil, I'm Brazilian, and I have developed some skills mostly in biometrics. So I tend to see myself as a plant maize, as where my roots are mostly in biometrics and statistics. And since my undergrad studies, I have developed some skills mostly for environmental modeling. And only in my master degree, I decided to get to the plant breeding field, where I learned more about quantitative genetics and at the time rice breeding in the University of Goiás and Embrapa, the Brazilian Corporation for Agricultural Research back in Brazil. And I tried to start to combine the environmental modeling and quantitative genetics. So I started to study this field. I have found very good papers already published for many research groups around the world. And then I went to University of São Paulo, also in Brazil, to have my PhD in quantitative genetics and maize breeding. And during this time, I spent some time in CIMMYT with José Crossa to learn more about genomic prediction. This gave me also the visualization of how Envirotyping, now we can also start to say the terminology of Enviromics, could combine with genomics to increase the accuracy and also to optimize the multi-environmental trial networks for genomic prediction. Since the last year, I came to Cornell to stay some time with Ed Buckler in his group. And my goal here is to start to study more about adaptive allele mining using Enviromics for this purpose. But also I'm doing some research about how we can increase the mechanistic view of GBLUP modeling using genome to fields data. So I will start this project yet. So because of this, today I'm going to focus more in my later research. The outline of this presentation is focused on genomic prediction across multiple environments. The goal of this field is to increase the GBLUP prediction across multiple environments, which involves actual environments you have already tested in the past or future environments. But for doing this research in the last few years, we faced some roadblocks. And we see that there's different aspects to be considered before starting these pipelines for genomic prediction with Enviromics. And some questions came up. For example, does environmental data really helps in these fields? Because we have found some papers that people compare models with and without environmental data, and sometimes they achieve almost the same accuracy. Also, we have a lot of kernel methods to implement this Enviromic genomic prediction. Which the best one? And sometimes we see negative predictive ability values. So the models are incapable to really predict the phenotypes. Why this happens? And we can predict future GBLUP. So this is gonna be very useful for plant breeders because we can start to use historical data sets to predict future years. So for example, we can optimize field trial and test efforts. We can start to predict climate change effects for different regions. So this is very important for plant breeding. But most importantly, all of these pipelines involving Enviromics, they are breeder friendly. They are easy to work. They are cheap. So we start to ask some questions, and we came for these three aspects. Mostly the ugly aspect is this is a not fresh field. A lot of people have suggested to use environmental data in plant breeding, and mostly more recently in genomic prediction. But it is still underused for most of the seed companies, and also mostly in developed countries that people don't have ways to collect the environmental data because it's very expensive. The sensors to put in the field, the weather stations, the soil sensors. So this is very expensive for developed countries. And also, some people start to ignore and treat this as no relevant. So we came with these questions, but the first bottleneck of this field is, okay, we have environmental data, but there's different equipments, there are different tools, there are different data sources. So how you connect these data sources, because they have difference in the measurements, and also what's the scale of this data? Is daily scale? Is across development stages? So we start to ask some questions and see that there is no clear pipeline to help breeders and help quantitative geneticists to establish these pipelines for Envirotyping. But most importantly, there is no public pipeline, and this is key for developed countries. As I told, it's very expensive to put the sensors in the field. But if you're thinking about this, also there's a theoretical gap in this field, because if you look for how environmental data collected in the field, this environmental data goes to some processing, the phenotype adjustment, statistical analysis, to get the actual phenotypes you're going to work for your studies. Molecular data is also collected in the field, goes to some quality control and genomic assembly and everything. So a lot of steps to achieve the actual markers you're going to be using for your studies. But environmental data, mostly people take the data from the weather stations and put it directly in the model. So there is no way to process this data, nor clear way to process this data. And this is very important to establish a very good way to use environmental data to actually increase the accuracy of genomic prediction and the other Envirotype pipelines. That's why today I choose those two papers from my PhD thesis. This paper here is about the package I developed with partnership with CIMMYT. It's called Envirotype. It's available here in GitHub. And also this proof of concept published last year, where we suggest a new way to create an environmental relatedness matrix, a new way to process the data, actually. So some good aspects about this. We across the years, have concluded that a good Envirotyping pipeline can be started using public and remote data sets. We have now a wide number of public data sets that covers the whole globe, and based on remote sensing. So these data sets are sometimes more or less accurate, depending on the data set. But for some places that we don't have any environmental data, they work. So we start to looking for these data sets, mostly the NASA Power and the World Clean and other data sets, soil grids, for example, and start to create this way to thinking. We have our environmental sensing step that we can involve actual or simulated environments. You can simulate new regions, for example. You have to process this data. This is key. So this processing of data involves the data control, but also a way to extract the biological information from this data. And one way we suggest to use for data processing is to create typologies, that is environmental types. We can, for each environmental factor, create a discrete values or bins, you can call, for each, using some ecophysiological knowledge to create these classes. And I will talk more about this in the future slides, but you can start to compute these discrete classes of environmental factors and then see the frequency of these factors for each environment. And then we can start to look for the environmental similarity as the shared frequencies across the environments. But before talking about environment, you must thinking about what is really environment. Because if you ask this question for different people, for different fields in agricultural research, they will get a different answer to you. Most people associate environment with location or a growing condition or a field experiment. So essentially, an environment for agricultural research is a combination of a certain geographic location in a certain planting date, a certain time development, crop development time. For example, a same location may face different environments according to different planting dates. And also, if you put different managements, this will affect the balance of resources in the environment. So this give you a new environment. For example, same location, same planting date, but different nitrogen levels. It's going to give you a different environment. So in this sense, we're going to start to think that environment is just a balance of a resource. And a good or bad environment will depend on how the plant will absorb and use this resource. Thinking also about environments in a global way, this is a very dummy example from the package. I took different places in the world and I played, okay, this is summer season. I took only one summer season for it and simulate the maize growing conditions. So you're gonna start to, for example, I take here my hometown in Brazil and some city very close, but other places in the world. And I take, for example, only the temperature and create these typologies, these classes of temperature. This is Celsius temperatures. And then we can start to plot these typologies and see how similar are the environments based on the shared typologies. A very interesting result here, for example, is that for this example I created for the package, in this year, in this specific year, the summer season in Texcoco and Brisbane, had almost the same environmental pattern for temperature. And also Palmas here in Brazil, almost the same pattern for Davis in California. So two places very close, that is Goiânia, my hometown here, and Palmas, they have very different environmental patterns for temperature if you consider an environmental combination of planting date and location. So geographic near locations are not necessarily have the same environmental pattern. And then you can start to take this matrix of frequencies and create a matrix of similarities. Here is a Gaussian kernel matrix that ranges from 0 to 1, which means that 0 is no similarity and 1 is maximum similarity. And you can start to group environments based on these similarities. In the package, we have functions called getWeather to collect this environmental data remotely, Envirotyping to create the typologies, and Envikernel to create the kernels or the matrix of similarities. The package offers a wide number of environmental covariables. Now, in the current version, we'll have more weather data and some elevation data, but I'm planning to put in the package a function called getSoil, for example, to collect the soil from soil grids. So you're going to expand the range of environmental data in the future months. But for now, we have the collection of this data, and we have some functions to process the data and create additional covariables. In terms of the covariable, for example, is F radiation use efficiency. That is a covariable I took from crop growth models for maize that corrects the effect of temperature on the radiation use efficiency for maize. So this is a covariable that ranges from 0 to 1 and means that if you have heat stress, this is going to affect the radiation use efficiency, for example. There's very nice covariables with a real biological meaning. For example, the difference between precipitation and evapotranspiration. So you can start to easily create these covariables using the package. I took as an example a data set you probably know very well. That is the genomic fields data from 4 years. And for example, for downloading this data, we have the following requisites. We need to put a time window, and this time window must be between 1981 and until yesterday. So you can download the environmental data from yesterday. Geographic coordinates, and the basic are skills and internet connection, obviously, because otherwise you're not able to download the data. And here, for example, is a very dumb example where I downloaded this environmental data for all these field trials in 2 minutes using the getWeather function from the package. Data processing must involve, in this example I will give to you today, some idea that the environmental factors must be grouped in typologies. And these typologies has a biological meaning behind. If you look for the Shelford law or the tolerance limits law from ecology, you can see that, for example, some species has a dispersion across an environmental gradient, where the region where the species has the more occurrence is actually the region with more adaptation, environmental adaptation. And the regions where the species are not adapted, probably you'll be regions where the environmental factors are very stressful and led to death. So if you're thinking about every species and every genotype actually has this distribution for across an environmental gradient. And this limits is species specific, but also can be adapted by breeding. But using this plot as a theoretical model, we can see that we can divide a distribution of certain environmental data in classes or discrete classes of beans or beans, for example, of environmental data. For example, the occurrence of intolerance by lack of this input, the occurrence of stress by lack of this input, the occurrence of optimal zones for this input, and the occurrence of stress by excess of this input, and the occurrence of zones of death by excess. So we can start to regulate which is frost, for example, which is thinking about temperature, what is heat stress, what is frost stress. So we start to look for the environmental data in this sense, in terms of frequency of these classes. Thinking about GBLUP prediction, you can see that the GBLUP in field trials is a consequence of the environmental diversity. For example, if you take two genotypes, theoretical genotypes, and you have two different plasticity curves for these genotypes, because they are different genotypes, you can see that if you took the limits of the environmental network, I mean the lower values and the highest values of certain environmental input, for example, temperature, you can start to plot some regressions over this environmental gradient. Some people call this reaction norm, but actual reaction norm is when you have a controlled condition, just like a globe chamber, you know exactly the amount of temperature you can control and the response of the phenotype. But in natural environments and also in field trials for plant breeding, we don't have any control about this gradient. But okay, you see in your phenotypes, these patterns here is actually the GBLUP. But if you change the experimental network, for example, another year, you're gonna have different environmental occurrences in this year, which will lead consequently to different GBLUP patterns. The same for another year, for example, and the same for another year. So the conclusion here is which drives the GBLUP patterns is the diversity of the environmental conditions, not the number of environments. And this is very important for predictive breeding because, for example, if you have this year of data sets and you train a genomic prediction model and try to predict different years, perhaps in this case, you're going to be very successful because we have a broad environmental diversity that covers the other multi-environmental trials, the other experimental networks across different years. But if you have this year, for example, and try to predict another years, you will not be able to predict the future or predict a different region because the GBLUP pattern is different. So before designing an experimental network, you must be aware about the diversity you expect to see. And as I told, rationalize is a consequence of the environmental diversity. GBLUP is a consequence of the reaction norms. GBLUP is multi-environmental trial specific. So if you change the number of locations, if you change the group of locations, you're going to change the GBLUP. But typologies are universal because if you break the environmental gradient in typologies, you can start to compute the frequencies no matter which year you're going to have. And for example, here you can start to see a broader range of frequencies of different typologies. While here you can start to see a small, short range of frequencies, but the same typologies, but in different frequencies. So typologies are transferable across years, for example. That's why characterizing typologies is key. But now I'm going to talk to you about the second concept, that is the difference between environmental characterization, envirotyping, and environments. In fact, environmental characterization is a very old concept that must cover or not the use of environmental data. Plant breeders use it, for example, the phenotypes for group environments based on the phenotypic value. This is a concept back in the 60s, for example. Back in the 90s and the early 2000 years, we have faced some papers using, for example, crop growth models to model the frequency of occurrence of the environmental factors, just like drought or heat stress or frost strength. This is the basics of Envirotyping, to see the frequency of certain environmental factor. And that's why Envirotyping is different for environmental characterization, but environmental characterization must involve, could involve Envirotyping or not. Enviromics, by the other hand, is the big picture that involves the different levels of the species. Since experimental networks, we can start to look for the environment of certain experimental network, but also look for a certain germplasm across years, for example, or a certain species across the globe, or different species sharing the same environment. That's why Enviromics is a big picture about, but always involve environmental data. I'm going to talk to you about Enviromic-aided genomic prediction. This is a terminology I'm using here to avoid the use reaction norm for genomic prediction, because as I told you before, reaction norm assumes that it has an established relation between the phenotypes and the environmental gradient. And here we're going to put environmental data in different ways in the model. So if you have a experimental network, we have this group of tested environments and tested genotypes in this purple square. If you try to predict new genotypes, you're going to borrow the covariances from these tested environments, but also using genomics to connect these expected new genotypes with the actual genotypes. But if you try to predict new environments, we have to borrow environmental data to correct these traits. And here, as I told you before, we can use typologies to connect different experimental networks. This also can help in predicting new GBLUP, because you can start to connect genomics and enviromics to emulate the future genotype by environment interactions. This is a very generic model. In the package, we can create this model very easily. We have two functions to create this model. The get kernel, where you put a list of G matrix or any other matrix for genetic relationships, must be additive, dominance, epistasis, whatever you want, and a list of environmental covariables, just environmental similarities, must be different environmental data or different development stages. You also are free to test different structures. And I also have this function, a kernel model, to use the outputs of get kernel in a Bayesian hierarchical framework. But talking very fast about this model, we have different structures here based on genetics, must be main genetic effects or GBLUP, not use environmental data. There's also envirotyping based kernels, that is similarities between the environments based on the envirotyping, and the combination of genomics and this envirotyping structure that some people call as a reaction norm, but is actually a representation of the reaction norm. So, the kernel model function was developed by Italo Granato. Now he is in UAE. I don't know if I think he's in France, but he was a former PhD from my lab back in Brazil. He's a very smart guy. He developed this function BGGE, and because this function is very optimized, so you can use a lot of numbers of different kernels very fast. That's why I'm using here this function. About the paper, we have some questions. The T matrix is suitable for prediction, so we're going to test this in this paper. The T matrix, as I told you, is a matrix of frequencies. This matrix is better than they use in the conventional way of environmental data, that is the actual mean-centered covariables. And also see that if you put, for example, this structure here, the kernel model for reaction norm, this really helps in the prediction of GBLUP, because sometimes if you have a very good environmental relationship matrix, this is going to combine with the genomics, cover the actual plasticity of the genotype. So we also test this and try to predict new GBLUP. This is a matrix of typologies. This is a fingerprint of the environments. Each row is a different environment, and each column is a different typology. We can start to look at this matrix as a fingerprint of the environments. Each entry of the matrix is a frequency of the certain typology in a certain environment. is with this matrix, we can start to create a relationship matrix between environments. In analogy with the matrix of molecular marker data by genotype, we create a kinship for using Van Hayden and other different kernel methods for creating genomic relationship matrix. We can start to use this matrix of environmental types to create kinships for the environment. Here we tested the different ways. I'll not get too much detail here, but we have developed a cross-validation scheme that we sample different number of environments and different number of genotypes. And here we plot the overall results. This is the actual number of environments by the actual total of environments we use. And in this figure, I want to call your attention to see that before in the x-axis here, we have the scenario here. We have a few phenotypes to predict a wide number of growing conditions. As going to the end of this axis, we have a different opposite scenario. We have a lot of phenotypes and also few new growing conditions to be predicted. And for example, if you look here, in the black is the GBLUP without Enviromics data. We see that this GBLUP is very stable, no matter how much phenotype data you use. But the two models use environmental data. The yellow is using the matrix of the W matrix, and the purple is using the T matrix. You can see that when we increase phenotypic information, the predictive ability drastically increases. But if you look to the bottom here, we start to see that in these conditions here, we have few phenotypes, okay, but we have environmental data. We start to see a really growth of accuracy. But if you have a very few number of phenotypes, which is selected by random cross-validation, we sometimes achieve worse predictive ability values. But I want to call your attention also to see that no matter which scenario we want to test, the T matrix always outperform the W matrix. And here, when we have a huge number of phenotypes, they have almost the same predictive ability, which means that the W matrix for environmental data always need a wide number of phenotypes. And here, there are some results interesting about this. This is actually the training set. I'm not talking more about this. But here, when we try to predict new environments, this is actually only the T matrix versus the GBLUP. We start to see that we have a growth of accuracy in the predictive ability. That's very good. But when we try to predict new genotypes or new GBLUP, we see this very weird performance. If you took only the median value of this distribution, they have almost the same accuracy. But when you look over the distribution, you see that sometimes they are very bad, but sometimes they are very good, which makes we to wonder, not every training set is useful for GBLUP prediction. And okay, if you have some good training sets, you can start to select the best training sets. That's why you came with this idea to use Enviromics to optimize field trials. I'm going to talk to you more about this in detail here. If, for example, we have a bank of inbreds or have already genotyped, we can start to create the hybrids in silico. So you have a matrix of hybrids only in silico, and have a G matrix for this. Using Enviromics, you can do the same. You create an environmental relatedness for a certain region you want to be tested, and combine those two matrices, and then use a genetic algorithm to select the best combinations of genotypes and environments. And then we start to see the actual best training set for genomic prediction, using less phenotyping efforts, but maintaining almost the same accuracy. And this is gonna be on one of the last slides. We have here a new concept about the resolution of genomic prediction, because sometimes people start to look at the model accuracy. But if you try to predict a new year, you can also start to correlate the performance of each genotype with its own performance in a theoretical environment, and start to see if the model represents the plasticity for each genotype. And here we test different model approaches after optimization. And after optimization, you can see that less than 10% of the multi-environmental trials are enough to predict the whole multi-environmental trials. And here, for example, the GBLUP has a very poor resolution. It's only capable to predict well less than 20% of the population. But when you put any environmental data after the optimization, no matter which model, is very good. So we conclude that, okay, the optimization using the T-matrix really helps no matter which Enviromics model. But the interesting thing here is when you try to predict the variations between environments, not the genotypes, but environments in the future year, the GBLUP is incapable of doing this. But when you put the T-matrix, it's very suitable to predict a new year. So you can reproduce the environmental diversity using the T-matrix. Finally, when you start to fit some Finland-Wilson regressions to mimic the linear plasticity of the genotypes, and we compare the reality with the predictions based on the phenotypes, you can start to see that this is the reality in the field trials. When you try to reproduce this reality using the GBLUP outcomes, that's not able to predict. They predict very, very, very bad, in a higher error. But when you use the T-matrix, this increases a lot. So it's not perfect yet, but increases the way to reproduce the plasticity. So the message here is plant breeders can also start to use the inbred lines bank of genotyping data and start to think in different regions they want to grow and test these future hybrids from these inbreds. Then can combine in silico those two matrices and then design the field trials for the future. And using these field trials optimized, they can also predict the whole population. So this is the message of this paper. And I think I'm out of time now, but this is the last slide about my conclusions. Yeah, environmental data really helps. To the best of our knowledge, really helps. I'm not able to present today, but we also published some results testing the nonlinear ways to create the environmental relatedness. And this is also not from my previous group, but these guys achieved almost the same result we achieved. That nonlinear kernels always perform better than the linear kernels. So sometimes if you are trying to predict a model with Enviromics and you are using a linear kernel, sometimes you have poor results because not of the environmental data, but because of the kernel method used to create the environmental relatedness. Predictability is not always bad. Is a suggestion that you can remove some bad phenotypes from your training set. So you start to think that Enviromics not only use it for characterizing environments and also increase accuracy, but also for optimizing multi-environmental trials. And yes, you can predict the future in a reasonable way using the T-matrix, because this is very interesting because it connects different multi-environmental trials and also natural environments. And after we finished the Envirotype, we came to the idea that yes, now it's very cheap to use environmental data if you don't have field sensors, and you're going to improve this package more. And we are glad to hear some feedbacks about this package because we always can improve the package for a broader audience. Finally, this is only a slide about my current project here in the lab. I'm going to start to work this project actually. But I'm going to bring these concepts about the Shelford's law and the diversity of the species for the Enviromics, and also start to use the typologies to understand how the different species are adapted for different regions. This is going to be accomplished with the environmental GWAS studies. This is for the Anthropogony tribe. And you can start also to think that we can create different similarities according to different environmental factors and see which is the most prone similarity to explain the species diversity. So I want to acknowledge my Enviromics network. These are people who teach me some about Enviromics and help sometimes. Special thank you to Alexandre Bryan, who teach me about crop growth models and Enviromics a few years ago, much before people say Enviromics terminology. My former advisor back in Brazil, in Universidade de Goiás, João Batista Duarte. My former PI from my University of São Paulo, he went to Erie, so I was his last PhD student, and now he's going to Louisiana to be a PI in there, so he's moving a lot. And Jose Costa, who helped me a lot during my PhD because I went to Mexico to have my internship, And 2 weeks when I arrived in there, the pandemic started, and I had to leave inside of the CIMMYT's headquarters during the pandemic. So without his help, I was not able to finish my thesis. So a special thanks for him. And also a thanks for the Berkeley Lab who received me last year, and I'm very happy to be part of this lab, and thank you so much for it. And now I'm open for questions. Thank you.